Hello, everyone. Good morning, good afternoon, or good evening. Welcome to the first pre recorded reinforcement lecture in clinical bacteriology. But before that, let me introduce myself. I'm Kate Lim. You can call me Miss Lim or Miss Kate, but most of the students prefer calling me the latter. And I will be your clinical bacteriology lecturer for today. So, before we get started, let me introduce to you microbiology. What is microbiology? What is the difference between microbiology and medical microbiology? Microbiology is the easier one. It is intended more to high school students and biology students, while medical microbiology, on the other hand, is for medical students and doctors. The difference between them is so huge and it's not even funny. Microbiology is like learning how to do the simple linear math equation, while medical microbiology, on the other end, is like learning how to fly a freaking airplane. It's scary at first, but at the moment you've learned how to do it, the possibilities are endless, and you maybe become a top gun. But not everyone gets to be a top gun. That's why you will be needing your goose, your wingman. So don't you worry, I'll be your wingman together with the rest of the bacteriology team. So now let's get started. At the end of this discussion, the student, you, should be able to define microbiology, pathogen, non pathogen, and opportunistic pathogen. Differentiate between acellular microbes and cellular microbes. List several reasons why microbes are important. Explain the relationship between microbes and infectious diseases. Differentiate between infectious diseases and microbial intoxications. Discuss two medically related fields of microbiology. And list some of the contributions of these awesome people like Leeuwenhoek, Pasteur, and Koch to microbiology. Differentiate between biogenesis and abiogenesis. Explain the germ theory of disease. And last but not the least, outline Koch's postulates. So, you ready? Microbiology as a science. Microbiology, in here you will meet a lot of creatures, like creatures, microorganisms. So these microorganisms may be gross and terrifying, while others are simply amazing. So get ready to discover the bacteria that makes you fart, the mite that lives in your eyelashes, and the fungus that turns ants into zombies. So, what is microbiology? Microbiology. Micro means small, bio means life, and logy means the study of. Although bio is referring to living organisms, microbiology includes the study of certain non living entities. Again, microbiology includes the study of non living entities. So, microbiology is the study of microscopic organisms, both living and non living. So, these microorganisms are collectively known as the microbes. Again, micro means small. That's why it must be viewed using the our best friend microscope. So the microscope. So these microbes are absolutely everywhere, meaning it's ubiquitous. It could be around you, on you, or it could be inside you. We live in harmony with some of them, and we're at war with the rest. So some of these microbes that I'm talking about are the virus, the bacteria, the archaea, protozoa, algae, and fungi. Generally, we divide these organisms or microorganisms rather depending on their general characteristics. As for the virus, we depend on its genome. It could be an RNA or could be DNA, whether it's be single stranded or a double stranded. And bacteria, on the other hand, we use the gram staining. Gram staining reaction could be a gram positive or a gram negative bacteria. And fungi, they are divided into molds, which are multicellular, and or yeasts, which are unicellular. So we will be talking more about these microorganisms, specifically bacteria, here in clinical bacteriology. So the microbes. Microbes are generally subdivided into two major categories. We have the acellular microorganisms or the infectious agents or the cellular microorganisms. Under the acellular agents or infectious particles or agents are the prions and viruses. Remember the definition of microbiology? What is again the definition of microbiology? Microbiology is the study of both living and non living entities. An example of a non living entity is a virus. Scientists do not consider viruses to be living organisms. Why? Viruses are not composed of cells. By the word itself, acellular, a means absence. So these microorganisms that are considered as acellular agents are not composed of cells at all and are considered infectious particles. Say, for example, for the virus. The virus consists of nothing more than a nucleic acid plus a protein coat. This protein coat is known to be the capsid. How about the prions? Prions are infectious protein molecules that causes diseases to both animals and humans. So, if viruses are composed of a nucleic acid and a protein coat, prions, on the other hand, are just infectious protein molecules. Infectious protein molecules that causes animal and human diseases. I will be introducing to you another term. If prions is composed of an infectious protein molecules, we have also the viroids. What are viroids? Viroids. Viroids. These are infectious RNA molecules that causes plant diseases. Viroids are infectious RNA molecules that cause plant diseases. So we have again, in summary, for acellular meaning absence of cells, or these microorganisms are not technically composed of cells. We have the prions. What are the prions once again? Prions, infectious protein molecules. We have the virus. They are composed of a nucleic acid and a protein coat named capsid. We have also the viroids, viroids. These are the infectious RNA molecules. So we're done with the acellular infectious agents. We're down to the cellular microorganisms. So if acellular infectious agents are not composed of cells, cellular microorganisms are, guess what? They're composed of cells. Cellular microorganisms are subdivided into two major categories. We have the prokaryotic and then the eukaryotic. Prokaryotes, we have the archaea and the bacteria. Archaea, bacteria, prokaryotes. Eukaryotes, on the other hand, are the algae, fungi, and protozoa. So let's just remember prokaryotes, archaea, and bacteria. What are prokaryotes? Prokaryotes are technically less complex or less complicated compared to eukaryotes. Why? Because eukaryotes has a true nucleus. By the word itself, eu means true. These microorganisms has a true nucleus and many membrane-bound organelles. That's why they are known to be more complex compared to the prokaryotes has no has no true nucleus. Again, prokaryotes are considered less complex, less complicated compared to eukaryotes. Why again? Because your eukaryotes has or contains a true nucleus, and a many membrane-bound organelles, while your prokaryotes doesn't have any. Don't you worry, I have a separate video lecture and video sessions about prokaryotes and eukaryotes, and we will be talking more about this on our clinical bacteriology journey in the next lecture sessions. So, so these microorganisms are also classified as non-pathogenic and pathogenic. So these non-pathogenic are those microorganisms that doesn't cause diseases, while your pathogenic are organisms that cause diseases. So I will be presenting to you some basic characteristic. So tell me if this characteristic is a non-pathogenic or a pathogenic. Number one, harmless. Harmless is a non-pathogenic. A microbe that causes infection and disease, a non-pathogenic microbe or a pathogenic microbe. Very good, a pathogenic microbe. A microbe that produces toxins, non-pathogenic or a pathogenic microbe. Good job, a pathogenic microbe. So easy, class, no? So right here, you can see the differences between the two. So we have the non-pathogenic and pathogenic by the word itself, non-pathogenic. So non-pathogenic does not cause or may not cause a disease to humans. So they're harmless and could be beneficial, and they live inside us or on us like the surface of the skin. Some of the non-pathogenic microorganisms are the saprophytes. What are the saprophytes? Saprophytes are the decomposers. So they live on the dead matter and they're responsible of decomposing the inorganic to organic matter. If you could just imagine if there's no saprophytes, diba? Yucks. So in here, non-pathogenic organisms are usually 70% of all bacteria. 70% of all bacteria are actually our friends, while the 30% our enemies. So these pathogenic or the 30% of bacteria causes infection and disease. They usually have variety of shapes. They produces toxins, they spread easily, and they live everywhere, meaning they are ubiquitous. So here is an example of a pathogenic microbes and the diseases they cause. So we have the algae. Usually, algae are cause rarely an infection. So they cause only when the humans ingest toxins. So usually through microbial intoxication. For the bacteria, we have a lot of diseases, and we will be talking each of these in our future lecture sessions. You have the fungi, allergies. Oh, take note, Tinea ringworm. It's a ringworm, but technically and scientifically, rather, it's not a worm or a parasite. It's a fungi. So ringworm are caused by fungi. Protozoa, oh, You have already discussed this during your parasitology class. So you're a master's of parasitology now. So you already know everything. You know all of these diseases if not familiar. Viruses, obviously, we have COVID 19, AIDS, and you will be talking a lot more of this during your virology class. So, for naming and classifying microorganisms, Carlos Linnaeus was so famous. We already knew him way back when we were in grade school up until now. So, who is Carlos Linnaeus? He was this genius who established the system of scientific nomenclature. He is a botanist and he is very curious too and a smart one of the smartest scientists ever be because he's the one who realized way back in 18th century that the botanical nomenclature of Europe was so complicated. So, in return, he developed the seven level taxa of classification system. Well, you know that the phylum, the kingdom. So he was the one who made that one. But don't you know that Carolus Linnaeus was got really famous when he started naming himself way back in the 18th century in his time. He only the rich people has surnames, and Carlos Linnaeus came from a very poor family, so he doesn't have a surname. So when he goes to college and when his classmate and his professor asked him, "What is your surname, Carolus?" So he just made up one, Linnaeus. It actually came from the linden trees that grow in his homestead. So he persevered and become a scientist and become famous up until now. So he introduced the binomial nomenclature. So binomial nomenclature, we have here the genus and the specific epithet. You have already been familiarizing this one ever since. So I will just be reiterating to you the most important ones. So here in the genus and specific epithet, just remember that both of these names should be italicized. The genus should be capitalized. And the specific epithet, the first letter of the specific epithet should be not capitalized. Uh, the names are usually Latinized and it is used worldwide. Latinized. Why? Just a trivia. I have read this somewhere. Way back before now, in 18th century, the researchers, the scientists, and aka the genius people, their basic language is Latin. So if you don't know how to speak Latin, then maybe you're not considered to be a genius back then. But again, scholars and scientists and researchers and even philosophers uses Latin as their main and basic language. So Linnaeus here, who is a very genius one, actually adopted a personal motto: God created, Linnaeus organized. Next slide here as an example of a species. We have a Staphylococcus aureus. Staphylococcus aureus. Staphylococcus is the genus name. Aureus is the specific epithet. Take note that this species name is italicized. The first letter of the genus name is in capital letter, while the aureus, which is the specific epithet or the second name, is not capitalized. So you could write the other way around. You could underline both the genus and the species if you don't know how to do the cursive writing. Just take note, class, that when during practical examinations or moving examinations in our core subject, the instructors will not allow the naming of S. aureus or E. coli, just while you putting shortening rather the name of the genus. So we don't allow such. So we need a complete name of a species during moving examinations. So again, these are some of the types of microorganisms. We have the bacteria, archaea, fungi, protozoa, algae, viruses, multicellular animal parasites or your protozoa, and the prions. So among here, class, I really had a fun time of categorizing this microorganism based on their um, what can I say? Intelligence? Yeah, intelligence. Right here, I actually categorized viruses. This is research proven, class. Viruses are actually considered one of the most intelligent microbe ever be. And it's quite obvious. Recently, we have experienced a pandemic that is caused by a virus. Viruses are considered to be most intelligent. Why? Because it has the capacity to adapt and readapt. It has the capacity to disassemble and assemble. They have the capacity to mimic a whole cell. So they're actually very intelligent, and that's the reason why in this modern and scientific world we had just experienced a pandemic that is caused by a virus. So upon knowing that, I really um, believe that viruses is one of the most intelligent microbe. Second in rank is the fungi. So fungi actually we rarely um, get this kind get some diseases that are caused by fungi because they have optimum temperature to be established. They need a minimum temperature to thrive and develop, and it's hard for fungi to thrive in just a normal temperature. But until this is my theory, this is my knowledge and my belief. Not until I've watched the Last of Us series. Have you watched that, class? You know, the Last of Us, the game. There's a series in HBO. I will be presenting this to you. Just a clip, a 1 minute clip of the first episode of the series. So, class, we lose. So, it's such a scary, scary place with these microbes. Okay, so bacteriology. Anyway, going back, The Last of Us series is a good series. So, if you have the time, you can watch it actually. Anyway, bacteriology. Bacteriology is the study of bacteria. So bacteria are made of just one cell each. So they're single celled or unicellular organisms. And they are considered the simplest cells on Earth. And don't you know that there are more bacteria on Earth than any other form of life? So again, bacteria are considered as prokaryotic, have examples like Eubacteria, Cyanobacteria, and Archaeobacteria. So they replicate through binary fission. So binary fission meaning they reproduce in one cell, splits in half to become two cells known as daughter cells. We will be talking more about this in our next lecture session. Mycology. The mycology, the um, possibility of having a pandemic cause of a fungi that is caused by a fungi is so scary, you know? So, mycology is the study of fungi. These are organisms whose cells have distinct nucleus, containing the cell's genetic material surrounded by a nuclear membrane, meaning they have a true nucleus. They are known to be eukaryotes. Again, fungi is subclassified according to their cellularity. It could be a unicellular, like your yeast, or multicellular, like your molds. So it could be monomorphic or dimorphic fungi too. They reproduce through budding or mitosis. We will be talking more about your fungi in your mycology class. The study of viruses is virology. So viruses are very small that can be seen only with the use of electron microscope. So they can't be seen in an ordinary microscope. So we will be using the electron microscope. What is electron microscope? The mechanism of an electron microscope is so easy. So it's just firing the tiny particles called as the electrons, or known to be the electrons at the virus, and then the electrons help make a computer picture. That's why we have a beautiful picture of a virus, because of our EM, or our electron microscope. So again, viruses are categorized either to be DNA or an RNA. Could be a single stranded or a double stranded. So they do not contain structured elements. So they are not considered as cellular microorganisms, and they had has the capacity to replicate inside the host cells. Say for example, we have the bacteriophage. By the word itself, bacteriophage, phage means eat, bacteria means bacteria. So what? An eating virus eating bacteria. So this is a bacteriophage. Some of these we will be talking more on our next lecture sessions. But you will be talking viruses in your virology class in detail. So parasitology, you're done with your parasitology class already, so I expect that you're already a master of these parasites. So parasitology is study of parasites, their host, their relationship, their mode of transmission, the life cycle, and their pathology. So examples of A parasite are your protozoa and your helminths. Your helminths is subdivided into three, if you remember. We have the nematodes, also known as, what is the common name of nematodes? Roundworms. How about the cestodes? Tapeworms. How about the trematodes? The flukes. Very good. So, why do we need to study microbiology aside from the fact that you need to pass this course? Why study microbiology? Normally, in my face-to-face classes, I have them write uh, the reason in a piece of paper and then study it with a pair. So it's impossible for us to do that now. So let's jump of the reasons why you need to study microbiology. So microbiology, the microbes that live on and in the human body are referred to as our indigenous microbiota. So, microbes, some of the microbes, if you remember, 70% of the bacteria are actually beneficial to humans, our friends. We live actually in harmony with them. So, indigenous microbiota, so these are the bacteria that lives inside us, protecting us from pathogenic organisms. So, indigenous microbiota is the new name. The older name of indigenous microbiota is the normal flora. But we don't use normal flora. We discourage the use of flora because flora refers to plants and microbes are not plants. So, this human microbiota, we have other names like indigenous microbiota, human microbiota. We call also them human microbiome or biome. So, most of the times they're of benefit to us. So, they help us or aid us in digestion, like the E. coli. So, there are some of them aid us in the food supplies, the secret materials. We secret some enzymes. You will know a lot more of these bacteria in our next sessions. Opportunistic pathogens, opportunistic. They do not cause normally a disease in ordinary conditions or in normal conditions, but if they have the opportunity to cause a disease, they will grab that opportunity and cause a disease. Opportunistic. But the word itself, opportunistic, mga opportunista. So it does not cause disease in under normal conditions, but if presented with the opportunity itself, they would cause a disease to a human or a healthy host. The human, usually it's not a healthy host because usually opportunistic pathogens um, cause a disease when the human is immunocompromised or the host is immunocompromised. Sorry, so it's not a healthy host. So that is your opportunistic pathogen. Example of an opportunistic pathogen is your E. coli. E. coli is an example of indigenous microbiota. So it helps actually in the digestion of our intestinal gut. But if your E. coli is dislodged and happen to dislodge in your urethra, so it is one of the causative agent of UTI. It may cause UTI. So that's opportunistic pathogen. Microbes are essential for life. Oh, see? Microbes are essential for life on this planet as we know it. For example, some microbes produce oxygen by the process known as photosynthesis. So, microbes, don't you know that microbes produce more oxygen than plants? So, microbes are actually beneficial too. Number four, many microbes are involved in the decomposition. So, these decomposers are known to be the saprophytes. So, decomposition, it is the process by which substance are broken down into simpler forms. So I have already discussed this to you, pre-discussed this to you that um, decompositions or the decomposers or the saprophytes helps in the decomposition process by converting inorganic to organic materials. Microbes are number five, we have microbes. They may be genetically engineered to clean after us, and this process is called the bioremediation. So genetically engineered microbes. We have a lot of researches rather, that that supports this kind of process, the use of microbes to that are genetically engineered to clean after humans. So number six, so algae and bacteria serve as food for tiny animals. So they um, serve as an important links in food chains. So food chains, don't you know that our food chains or the food supplies to humans are actually depleting or actually in danger. So some animals, say pigs for example, are getting sick, like the swine flu. So researchers proposed a new member of the food chain, so or a food supply to humans, and those are the rabbits. Yes, the rabbits. So s- actually rabbits or bunnies are consumed na in some western countries, but it's not a normal food for us here in the Philippines or in Asia. Some of us has rabbits, treats rabbits as pets. So it's such a very sad reality. Okay. Furthermore, we have number seven, microbes that lives in the intestinal tracts of animals. So they aid in digestion of food. So this is some example of human microbiota. So they help in producing substances that are of value to host animals. Example is the E. coli. So E. coli that lives in the human intestinal tract produce vitamin K and B1. Number eight, microbes are essential in various food and beverage industries. So these are used and utilized in the biotechnology. So like the beer and the wines, how they are used through the process of pasteurization, and for the fermentation we use the cheese and the kimchi and yogurt. So these are some of the examples of biotechnology. And some bacteria and fungi produce antibiotics to treat patients with infectious diseases. So technically, why study microbiology? Because not all microbes are dangerous or harmful. Some of these microbes are of beneficial to us and our friends and help us uh, live a more comfortable life. So, microbes usually causes diseases. So these diseases are categorized as an infectious disease or a microbial intoxication. I will be presenting to you some series of characteristics, and you will be identifying which characteristic refers to be an infectious disease or a microbial intoxication. So the toxin causes a disease. It is microbial intoxication. The pathogen causes the disease. It is an example of an infectious disease. So just take note the difference between the two. So we have different causative agents. For the infectious disease, we have the pathogen that colonizes a person's body. While in microbial intoxication, it is the pathogen which produces a toxin in vitro. What is in vitro? Meaning artificially made outside the body, in vitro, or it could be made outside the body, simply made outside the body, compared to in vivo, which means inside the body. So, in infectious diseases, obviously, the pathogen is the main causative agent of the disease, while in the microbial intoxication, it's the toxin that causes the disease. So in here, we have infectious diseases, causative agent, usually is the pathogen itself, and the microbial intoxication, the causative agent is usually the toxin that is usually ingested by the person. Okay? So easy. So right here, how about this case scenario? Patient X was admitted due to abdominal pain. Workup was done and revealed toxin ingestion. So what is your answer? It is an infectious disease? Just kidding. It's microbial intoxication. So your word here or your clue here is the toxin. The toxin is the causative agent for microbial intoxication. So medical and clinical microbiology. Medical microbiology is again the study of pathogens, the diseases they cause, and the body's defense against diseases. So, a subspecialty of medical microbiology is the diagnostic microbiology or the clinical microbiology. So, this concern more with diagnosis, laboratory diagnosis in specific. So, this is where medical technologies came in, the laboratory diagnosis of some infectious diseases that is caused by the bacteria to humans. So, we're down to the next part of this lecture session. How are you feeling? Okay, so let's proceed to the pioneers of microbiology, the history. When I was in college, I really hate history classes. But when I become an adult and become more mature, I appreciate more these people because without them, we don't have any vaccines, and maybe without them, maybe we're already dead because caused by some of the diseases. Mm. So let's play guess who of these people, of these awesome people. Who is this Mister who discovered the foundation of cell theory? This Mister is Robert Hooke. So, Hooke's discovery led to the understanding of cells as the smallest units of life. So, he was the one who introduced the cell theory. So, he is also the one who says that bacteria are our ancestors because they were the first living cells to appear on Earth. So, how about this guy? This guy is the father of microbiology, the father of bacteriology, and the father of protozoology. This Mister is Anton von Leeuwenhoek. So Anton von Leeuwenhoek is the father of bacteriology, protozoology, and is considered as the first true microbiologist. Why? Because he was the one who observed and accurately described living microorganisms. He even named these microorganisms as animalcules. Why animalcules? So unscientific, no? Don't you know that Anton von Leeuwenhoek is not a scientist? He was a fabric merchant, a wine assayer, a minor city official, and his hobby was just grounding tiny glass lenses and mounting it in a metal frame. And this metal frame is the single lens microscope. So this is the creation of the single lens microscope. His self-made single lens microscope has a magnification up to 300. It could magnify objects up until 300 to its size. But his famous work of art was lost at his death because he had not taught anyone the skill. So he's actually greedy at that part, but he was so curious because of his curiosity, he was tagged and named to be the father of bacteriology, protozoology, and considered as the first true microbiologist. So, right after Anton von Leeuwenhoek discovered the previously invisible world of microorganisms, so the scientific community of that time became interested in the origins of these tiny living things, or as Anton von Leeuwenhoek says, the animalcules. So they become very curious until the second half of the 19th century. Many scientists speculate on the origin of these animalcules or of these living things. And some philosophers believe that some forms of life could arise spontaneously from non living matter, and they call this hypothetical process as spontaneous generation. So, in this transition period, we have lots of debates between spontaneous generation or the abiogenesis versus the biogenesis. So, again, what is a spontaneous generation theory or the abiogenesis? So, abiogenesis is a belief that living organisms arose or arise spontaneously from non living decaying organic matter. So, people actually believe that toads, snakes, and mice could be born of most of moist soil, that is moist, of moist soil, and flies could emerge from manure, and maggots could arise from decaying corpses. Imagine that one. And this kind of mentality and thinking took another 200 years to disprove. So some of those people really actually believe that toads emerge from a soil, or maggots could arise from corpses, that living organisms arise from non-living matter. Diba? So, if you could just imagine now, class, or recall that one belief that you truly believed in, if you could think of that thing, can you imagine 200 years after, maybe that thing that you truly believed in is may not be true? So it could be, right? At that time, they truly believed that living organisms arise from non living matter. So this took a long 200 years to disprove. A lot of debates, actually. And the first uh, person to propose this kind of theory is Aristotle. Just imagine, class, BC 384 to 322 before Christ. So they believe that Aristotle believed that living things could develop spontaneously from non-living materials. So he supported this fact by saying that living creatures actually be found in some decaying meats. So this proposal of Aristotle says that non-living materials, life arise from non-living materials if the material contain pneuma. What is pneuma, class? Pneuma is breath in Greek. In religious context, it means soul or spirit. And science, it means vital heat. So Aristotle states that living things could develop or arise from non-living materials if that non-living material has pneuma, or has breath, or has a soul, or has a spirit. Diba? How foolish that mentality is. So that was Aristotle. But don't you know, class, that Dante from the Divine Comedy, if you have read his literature, he describes that Aristotle is the master of men who knew, because he believes that nature had revealed its greatest secrets to Aristotle. So Aristotle was considered one of the most brightest and smartest philosophers. But that was on 384 322 BC before Christ, up until Francesco Redi. Francesco Redi introduced an experimental procedure. He wants to disprove Aristotle's thinking. That is in 16th century. So he challenged the idea that no, maggots do not arise spontaneously from decaying meat. So he conducted an experiment. Right here, class, as you can see, three jars. But the first or initial experiment of Francesco Redi has only two jars. So in his first experiment, we have he conducted an experiment of the decaying meat. So this decaying meat is placed in an open jar, and another decaying meat to a tightly sealed jar. So Reddy just left the two jars for a day or two, and then discovered that the open jars has maggots in it. So he observed that flies that that which flew around the decaying meat lay eggs on the decaying meat that later on developed to become a maggots. While in tightly sealed jars, he discovered that no maggots were formed because no flies can come in contact with the decaying meat. So, right after this experiment, he presented this to the people and to some scientists as well. But Francesco Redi has a lot of antagonists and bashers, and his antagonists were not convinced. They claimed that fresh air was needed for spontaneous generation. You know what? Aristotle says that we need pneuma, we need air, we need breath. So he set up a second experiment in which three jars were covered with a fine net, in which the third jar, rather, was covered with a fine net instead of being sealed. But then again, no larvae appeared in the gauze that covered the jars even though the air was present. So maggots appeared only if flies were allowed to leave their eggs on the meat. So the work of Redi was a serious blow to the long held belief that large forms of life could arise from non life. However, many scientists still believed that small organisms were simple enough to be generated from non living materials. So, in summary, at the end of the day, they neglected and rejected Reddy's experiment. So, up until we have John Needham. So, John Needham supported Aristotle's experiment. He asserted that organic matter possessed a vital force at iya class that could give rise to life. So, this experiment is consisted of briefly boiling broth mixture and then cooling the mixture in an open container. Take note, class. In an open container to room temperature. Later, the flask would be sealed and microbes would grow a few days later. So these experiments seem to be a show that there was a life force that produced spontaneous generation. So this experiment shows daw class nga naa life force or a vital force that could give rise to life because of the microbial growth. Not until Lazaro Spallanzani disproves John Needham's experiment. So he improved the previous experiment of Needham by heating the broth placed in a sealed jar. So he proposed that the air carried microorganisms to the culture medium. So what he did, he heated the broth. Right after heating the broth, he sealed the jar with the broth in it. So after sitting it for how many hours, he observed that there is no growth of microorganisms in the jar. But despite that, still, some scientists neglected and rejected that idea until this theory of biogenesis was introduced by Rudolf Virchow. So, theory of biogenesis. This states that living cells that can arise from pre-existing living cells. So, this is the theory that um, that Rudolf Virchow introduced. So, he challenged the doctrine of spontaneous generation with the concept of biogenesis. So Virchow's theory stated that just as animals, just like here, like eggs and chickens, they are unable to arise without previously existing animals, and cells are unable to arise without previously existing cells. And during the 18th century, this was supported by Louis Pasteur. He was really the one who disproved the doctrine of spontaneous generation. So he proved that the air does not generate itself and microorganisms are indeed present in it and can contaminate sterile solutions. What what is a solution or sterile environment? Sterile means absence of microorganisms. So he proposed the aseptic technique. Using the heat in killing microbes, this is a method used in preventing contamination by unwanted microorganisms. Aseptic, meaning septic. What is septic? This is septic or sepsis. This is the invasion of microorganisms or pathogen inside the body, diba? But in septic, so presence or the presence of microorganisms. So when we say aseptic, the absence of microorganisms. So aseptic technique is established and could be done by the use of heat. So he provided evidence that microorganisms could not originate from mystical forces. Say, hey Aristotle, what you've been trying to say is not true. So he provided that these mystical forces is not present in non-living materials. So this is how he confirmed the his theory of biogenesis through his S-shaped swan neck flask experiment. So he heated an infusion. This in number one, as you can see in the picture, the nutrient broth is sealed in a vessel with S shape or swan neck flask here, as you can see. So the swan neck flask is very important in killing any microorganisms already present in the liquid. So this process, by the way, was later known as pasteurization. So again, he heated an infusion and then let it cool down and then broke off the tip of the vessel right here, as you can see in number two. This allowed this allowed the fresh air to enter. Diba yan sila na, oh, Aristotle says na we need fresh air. We need a pneuma. So he says, okay, I will let the fresh air to enter. So he let the fresh air to enter by breaking off the tip of the vessel. But any particulate matter that was present in the air was trapped in the bend of the neck of this swan shaped flask right here. As you can see in number two, here, one. So no matter how long he allowed the flask to sit, microorganisms never appeared in the infusion. So that was the beginning of the golden age of microbiology. With the help of his friend Theodore Swan, who also developed the process of fermentation. He described, Pas- Louis Pasteur described that certain microorganisms known as yeast has the capacity to convert sugar to alcohol in the absence of air. So Pasteur, with the help of his process known as pasteurization, he developed the wining, the the wine industry and the beer industry, and states that the souring of wine are caused by different bacteria. So he suggested that the minimal heating of beers and wines is enough to kill bacteria that can cause the spoilage. This is the process of pasteurization. So pasteurization was already used and still recently used in the winemaking industry and winemaking processes. So, Louis Pasteur's contribution to microbial science are the following. So, he disproved the theory of spontaneous generation. So, he is the one who proved Aristotle and the rest of the scientists wrong. So, without him, we'll be foolish enough to know or to believe that some living things arise from non living things. Thanks to Louis Pasteur, we disproved and debunked that idea. Number two, he developed the vaccines against anthrax and rabies. And number three, he improved the winemaking processes. We're enjoying our wines now thanks to Louis Pasteur. So we have here another two Nobel people who developed the theory of antisepsis. Ignaz Semmelweis, he demonstrated the routine hand washing. Take note, routine hand washing. So Ignaz Semmelweis. So Joseph Lister, he introduced the system of antiseptic surgery. He was the one who introduced the system of antiseptic surgery, and he was the one who introduced as well the use of phenol as an antimicrobial agent. So Joseph Lister introduced antiseptic surgery, while Ignaz Semmelweis was the one who demonstrated first the routine hand washing. So right here is a photo of just a photo of Ignaz Semmelweis. This is Ignaz Semmelweis right there. So Ignaz Semmelweis was an OB doctor. He was the one who introduced antiseptic prophylaxis. What is prophylaxis, class? Prophylaxis is prevention. So Ignaz Semmelweis was an OB. He has a very sad story, class. He was in his time in 1840s. He was the one he who discovered and observed that some of the um, female women who delivered their babies, who just delivered their babies, died because of fever or sepsis. So he discovered that these women were examined by student doctors who had not washed their hands after leaving the autopsy room. So when a colleague who had received a scalpel that got died of infection, Semmelweis called these doctors and ordered these doctors to wash their hands with chlorinated lime before examining patients. And as a result, the maternal death rate was reduced from 12 to 1%. So this idea struck Semmelweis, struck Ignaz. So what he did, he proposed publications. He have done a lot of researches in regards to this theory. So he proposed to his hospital that hand washing is a mandatory, should be mandatory, and should be done by all medical professionals. But despite of his research and his doings, the hospital neglected and rejected his studies. So Ignace broke down, he was depressed, and he was admitted to a mental hospital. In the mental hospital, he died of an infection. So that is the sad story of Ignace Semmelweis. So, proceeding to the person who saved humanity. It was Robert Koch. Robert Koch was the first person who showed an irrefutable proof that bacteria indeed cause a disease. So he discovered some causative agents like Bacillus anthracis, which is the causative agent of anthrax, Mycobacterium tuberculosis, the causative agent of PTB. And he was also the first one to cultivate bacteria in vitro. And because of that, he developed his postulates. What is his postulates? The first postulate of Koch was the suspected causative agent should be absent from all healthy organisms. So, causative agents should be absent from all healthy organisms, but present in all diseased organisms. So he conducted an experiment. So in number two, the causative agent must be isolated from the diseased organism. So it should be isolated and should be grown in a pure culture. So in number one, the suspected causative agent must be absent in healthy organism but present in diseased organisms. And number two, the causative agent that causes the death of a diseased organism should be extracted and should be grown in a pure culture. And number three, the cultured agent or the causative agent should cause the same disease when inoculated again to a healthy susceptible organism. In this case, in his experiment, we are using experimental rats. So in number four, after inoculating or injecting the cultured agent to the experimental animal, the same causative agent must then be extracted and re-isolated from the inoculated diseased organism. So these are the postulates, the four postulates made by Koch or Robert Koch. But there are still exceptions to Koch's postulates. What are they? So these are um, about the pathogens who cannot grow in culture media. Take note in your Cox postulates, it is a mandatory. He says that it's a mandatory that the causative agent or the causative agent that causes the death of the disease organism should be extracted and re-isolated in a culture media or in a pure culture. But then, some pathogens do not grow on culture media. Some examples are your obligate intracellular microbes. So, obligate intracellular microbes are also known as obligate intracellular parasitic microorganisms. Take note, class, that parasitic microorganisms and parasites are actually different. All parasites are parasitic microorganisms, but not all parasitic microorganisms are parasites. Parasitic microorganisms require a host to survive. So, best example of these parasitic microorganism is a virus, meaning they cannot thrive or they cannot grow. They cannot continue living without a host cell. So, some examples right here is the virus. We have the rickettsia, chlamydia, mycobacterium leprae, and some of the treponins. So, these are considered as obligate intracellular. So, they need a host to survive. Number two, some pathogens are animals. Some pathogens are animal or host specific, meaning mga pilian. They're specific. They cannot grow in just an ordinary animal or they cannot grow into a different host. Some of them are host specific. So they cannot cause a disease to an animal, but maybe can be infectious or be pathogenic to humans. So some of these pathogens are considered host specific. Number three, some disease may be caused by combined effects of two or more microbes. So this is what we know as polymicrobial infection. An example of a polymicrobial infection is the multiple sclerosis. Usually multiple sclerosis is caused by EBV and retrovirus. So some diseases are not caused by a single causative agent. Usually, some of them are caused by the two or more microorganisms. Number four, some pathogens may be altered when grown in vitro. Some may become less or non pathogenic. So, some pathogens are more pathogenic when they are inside a human host or inside the body. So outside or in vitro, they may be become less pathogenic or non-pathogenic at all. So some of Koch's collaborators are Fanny, Julius, Martinus, and Sergi. Fanny is the one who suggested the use of agar or the solidifying agent in the preparation of a culture media. In our next lecture sessions, we will be discussing more about the culture media based on their classification, based on their function, and based on their consistency. So this includes in the consistency, we have a solid agar, a semi-solid agar, a liquid gel um, media. So we will be discussing more about that later on. But it was Fanny who suggested the use of a solidifying agent, which is known to be the agar. And Julius Petri. So he will develop the Petri dish. Originating and developed class, this one, the Petri dish. And we have Martinus and Sergi. So they developed the enrichment culture technique. Enrichment culture technique. So later on, we will be discussing the types of culture media and the techniques of how to make a good and healthy culture media for the bacteria to thrive, because some bacteria class are very sensitive and very selective. So some bacteria require specific um, minerals and vitamins or supplements so they can grow. So Martinus and Sergi was the one who developed this kind of technique. So some bacteria or all bacteria could grow in this kind of media or culture. Next, we have uh, some of notable people in immunology. Let's just have a quick or brief discussion. You will be discussing this on your immunology class. We have Edward Jenner. Edward Jenner, he introduced the concept of vaccination. And Louis Pasteur and Pierre Rowe. So Pasteur used the term vaccine for an attenuated culture. What is attenuated? Meaning these are live organisms that are weakened or their pathogenicity is weakened. So some vaccines that are introduced to us are actually has a live organism or this live organism's pathogenicity are weekend. So this term is used to um, to produce attenuated vaccines. So Louis Pasteur and it was Pierre Rowe who discovered such attenuated cultures. And we are down to the modern therapy or the magic bullet. So, magic bullet, by the way, is in reference to an old myth about a bullet that cannot miss his target. Paul Ehrlich is a reader. So, he was the one who formulated this idea that it is possible, that it could be possible to kill microbes which causes these diseases in the body without harming the body itself. So it was Paul Ehrlich who discovered the salvarsan for the treatment of syphilis. So we have another magic bullet modern therapy applications. We have Waxman and Alexander Fleming. Salman is the one who discovered streptomycin and neomycin and regarded as the father of antibiotics. And Alexander Fleming, he accidentally discovered antibiotic penicillin through Penicillium notatum. He also discovered the lysozyme. So, we are done with all of the people, notable people, and some general introduction about microbiology and its classifications. So now, may I ask you, what career field in microbiology are you interested in and why? So some of these are some examples of careers you can have when you specialize in microbiology. You have bacteriologist, you could be a phycologist, protozoologist, mycologist, virologist, or medical microbiologist. Clinical microbiologist, that is the closest in us, for us, medtechs, or epidemiologist. So if you have chosen a career or if you don't want to choose a career in microbiology, that's okay too. What's important is you're happy with what you choose or what career field you are about to choose. I myself is not a practicing medtech. I had a clinical experience in a laboratory setting, in a hospital setting, years ago. But I chose to become an academe, to to be part of the academic team. So we have actually lots of choices, and I've been teaching for 7 years now. So we have a variety of choices. Don't enclose yourself in one field or no one career setup. So you could be maybe a physician someday, or if you don't want to pursue medtech in the future, but that's after graduating na ha. If you don't like being a medtech at all, you can pursue your hobbies as well. Maybe you could be a notable person too, and be the first of this to discover some things that we haven't discovered yet. Okay, so either way, that's it for today's lecture session. I hope you have learned something. I'm really sorry that my voice is kind of tired and shaky. I have been discussing uh for to a lot of classes already. So this is, by the way, my references. So it is Burton's in microbiology. I have already uploaded all the references to be utilized here in this course subject in your Google Drive. Kindly check it out. And this is a passage from Dan Brown. Dan Brown is one of my favorite author. I have read all of his books. So this is a passage from his book The Origin. May our philosophies keep pace with our technologies. May our compassion keep pace with our powers, and may love, not fear, be the engine of change. Again, worry ends when faith begins. So this is Miss Kate again, and thank you so much for listening. I will be seeing you soon. Thank you.